let me address the second one first quick. So we are kind of counting on an inflation-adjusted increase over time. And I think that's kind of what everybody would expect. But we cannot afford jumps from one year to the other to double, because we had 75,000 for maintenance, or to put it in half because we were in a year where we didn't have that expense. So what I really think what we need to be more proactive in, up till now, how we've dealt with it is we have written small grants. Some are university internal. We have also sometimes been backed up by the Center for Structural Biology to take care of some of these fluctuations. In the longer run, what we may have to do is we may have to introduce some depreciation on cheaper items and recover maybe 20 or 30,000 in depreciations that we can fold into the user fees that don't disappear at the end of the year. That's the key. The first question about the screening, we're basically working with groups that know how to do the screening. They do it on their own, and they propagate the information to other groups that might be interested. So we're really just providing the tools for them. On the IVDR screening, we are actually more the ones as a core who provide that as a service. I'm not sure whether that answers your question.
Is it like doing an experiment, or is it making a measurement, or is it to see if, in cryo-EM, there's target for like screening and data collection, and I kind of don't like the term, so I was just kind of curious.
So screening in our case, or with my definition, is really we have libraries of 10 to 15,000 small molecules. We have a target protein, and we want to expose the target protein to all 10 to 15,000 ligands, and basically measure either in combination or individually each of these combinations. That's how we call screening. Yeah. We don't. No, not yet. We've tossed around the idea a number of times, and to me, as long as the PI is willing to pay then the user fees, it's folded into that, and it's a service that we provide to increase our user base. So I look more at it from that perspective than as a revenue item.
Thank you. Any more questions we can hold to the end?
Good morning. Thank you for coming to hear all about structural biology. I have to say up front that I dabble in a lot of different cryo modes, so this is only one part of what I do or have done. So I currently am the director of electron microscopy for the University of Maryland, Baltimore. The School of Medicine and the School of Dentistry subsidize my deficits, which I run on a regular basis. So the fundamental tenet of biology is that we're going to study the relationship between structure and function. This is what structural biology does. What we're interested in is the macromolecules. They carry out most of the functions of the cells, and their 3D shape tells us a lot about how they perform their functions. So this session you're hearing from NMR folks, X-ray crystallographers, and cryo-EM folks, just to tell you how we get all the different information we put together to get a 3D rendering of the molecule in question. Cryo-EM does cool stuff. It allows us to study individual proteins and molecular complexes at a very high resolution. Cryo-EM can mean a lot of things to a lot of people. For this talk, it means single particle analysis, or SPA. There is volume EM, which we heard about at 9 o'clock from Kurt Zimick. There's electron tomography, there's high pressure freezing and freeze substitution, cryo ultramicrotomy, there's just, the list goes on. You can put cryo in front of just about everything you can do with an EM, and there is a instrument out there with a higher price tag that you can buy to do it. The method background for single particle analysis is straightforward, but yet complex and complicated because it requires an art. You're going to purify your proteins, you're going to flash freeze them in liquid ethane to preserve their native state of whatever that protein or molecule is when you freeze it. You're going to keep it cold throughout your whole imaging pipeline. Then you have all these pictures with this noisy background, just gray and white specks, and you have to figure out where your particles are and reconstruct a 3D structure from them. So a lot of this is done with alignments and classifications and segmentation and computers. As with everything, each cryo technique has its own difficulties and limitations. I tried to balance the talk between the positives and the negatives because when you're involved in it, you're so in the weeds that everything seems negative because everything seems to be a struggle. But in reality, when you go to the publications and the journals and the meetings, the data coming from single particle is really moving science forward. So single particle is becoming a go-to technique. It can do a wide range of molecular weights. It requires less sample than some techniques. It also allows you to deal with some sample heterogeneity, so it has some benefits. Essentially, you have that conversation with your core directors. You find out what it is that your project would benefit from. They ask you all these questions that allow them to delineate what machine they're going to put you on, what technique they're going to show you. Just sort of quickly, this was a whole set of questions that you sort of ask them. What sample do you want to do? Is it feasible? Is it pure? Is it homogeneous? Did they get their sample in a single layer when they vitrified it? So poor samples from the user are going to certainly impact what it is that you can do for them. And they need to understand that up front because it's not a one and done experiment. This is iterative. You learn something with every pass of sample making. So the sample's put on a sample grid, it's plunged in liquid ethane, you get vitreous ice, not crystalline ice, because crystalline ice is going to poke holes in everything. Those large crystals get in the way. The apparatus to do this plunging, expensive. So you need to have that. You need to have somebody who knows how to run it. Again, you've got that, there's some things that are automated, but there's some things that require that human touch, that human sixth sense about whether or not it was a good freeze. You then have your proteins in this ice, but there's a lot of difficulty because there's less contrast in them. So you have a hard time spotting your molecules, you have a hard time aligning them. So then you get onto the analysis. Once you've spent hours collecting all these images, heavy, heavy computer analysis is done here. So for instance, the ABRF2024 that I just had installed in the package was not the $20,000 image analysis computer that I needed. What I know is that there will be $5,000 every year from now on to update that software. So this is something that didn't come as part of the grant that the administration had to be willing to put up the money for, or in some cases, we're just going to increase my deficit. So basically for model building, you're going to apply averaging of the images. I told you you had a lot of particles in your frozen ice. They're hard to see. The computer's going to pick them out. You need to do a lot of particles. So you need to average these particles to pull out those densities that are going to help you to form your model. You're going to classify them. You're going to need to know something about statistics. Bioinformatics, statistics, a lot of places have these departments or these specialists. Many places don't, and you become the one-man show for whatever it is that your user needs to do in order to do their particle reconstruction. So it's not just about installing the unit, it's not about paying for the unit, it's about now they have all this data. If you don't help them make something out of this data, all that other effort is useless, because now you just have a hard drive full of data that they're not getting anything out of. So here's an example of one of these just very busy and full images of just gray. So how are you going to get your data out of this? The computers will help you, but you don't realize that when you read a journal article. So cryo-EM is gaining in popularity. The hardware, the software advances, all these things are helping to get higher resolutions, and so people see it in the journal and they come to you and they want to do this. There are numerous pitfalls and considerations that you don't see when you read a journal article. So this is where the pre-project meeting comes in, where I explain to them all of the different things that they need to think about, and sometimes go back and think about and redesign. But if I want a new camera, I need to think about this as an upgrade that's going to be very expensive, and how am I going to pay for this? Replacing aging technology is certainly something across the board that cores need to do, and it's very much out of the reach of what your user fees are bringing in. I threw this together just to give an example of a low end of what a cryo-EM core needs, okay? You need to buy the microscope. $5 million for a 300 kV cryo-TEM. That's straight out of the mouth of one of the vendors. I polled some of my fellow EM cryo core core directors, and I also had a chat with several people from the different vendors, just to get an idea of what the challenges were that they were seeing. And the biggest thing, amazingly, was the space. Once you have written that grant, once you have put in your purchase order for your equipment, or before really, you should think about the space. Where's it going? Your vendor will send you the specifics. So if you don't have a room, which most of us don't have these fancy rooms, then you need to have one renovated. That's not on my S10 grant. So you have to go to your administration and say, okay, you said you were going to support the grant. Now here's, this scope is coming. So you've got consumables to use it, you've got the service contracts once it's in, you've got how much are you going to pay your dedicated operator. You need somebody who understands this. We're not any longer just users and trainers of simple equipment. So fantastically enough, Nature Methods has a method of the year that they're going with. And this was news to me. I apparently don't pay attention to Nature Methods. Single particle analysis is a big deal, and it's been a big deal for many years. The Nobel Prize was given out for this. High resolution of structure determination of biomolecules in a solution. So how do you create your protein? How do you freeze your protein? How do you image your protein? All of these things had to be worked out. You needed your hardware, your software, preparation techniques. The whole pipeline needs to be worked out before you get that final 3D reconstruction. So explosion in the field, number of instruments, number of facilities, number of scientists, number of publications. Most importantly, the number of structures that are being deposited in the different databases. That's where NIH wants to see us going. Sort of quickly, you've all seen a little bit about this. If I show you a rabbit that I made with my fingers and a shadow puppet, it doesn't tell you anything about the molecule itself, and that's what we're trying to do. So transmission electron microscopy is a 2D image that you get, but we're trying to image a 3D thing. There's all kinds of stuff in the literature about how to do this. frozen samples are the best way. Software to align it, it goes on and on, it's iterative. The best thing for the field is to have those data sets sort of out there. The generated data can give you more data from other people asking other questions. So this data needs to be harvested, it needs to be standardized, all the metadata needs to be there. Then somebody else can go and look at your data set and start asking other questions. Does this molecule fit in? Does that molecule fit in? We'll pass over some funky, so these are the final images that we're all trying to get, right? 3D, what does the ribosome look like? What does the tau protein look like? What does the receptors on the SARS-CoV-2 look like? IT challenges, data handling, data management, data analysis. All this happens in the core. You've got to keep it up to date. You've got to purchase it. You've got to maintain it. You've got to move it. If you're lucky, like at UMB, we just hired an image analysis specialist. So I'm going to draw on their confocal experience to try to make my pipeline for all my volume data that's coming out of my ABRFO. You need to publish the data, you need to share the data, you need to understand that these databases have fees, they have restrictions, they have guidelines. You need to follow all of these things from the start so that you don't have to go back and redo your data. A lot of this computer analysis, speed, storage, all of this needs to go into the grants, the budgets. It has to be costed into the research. You can't any longer say, oh, I just need a $3,000 Dell computer to run my stuff. No, 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 no. So data management is a whole big thing. The volume EM world has created, through the Chan Zuckerberg initiative, a whole community with databases, listserver, courses, seminars, the whole 9 yards. It's becoming big. We're told do more with less. Okay, so as a core director, I'm tired of hearing that. A lot of us think that the core paradigm needs to shift. We need to, come on. So the complexity of the instruments, the samples, the data analysis has all changed. We're no longer a service center. We're now your collaborator. We have cutting edge equipment, or we want cutting edge equipment, and so now you need thought leaders to run that. And the institutions need to step up, and so the mentality of the administrators. I have landed in a very good place. UMB understands a lot of this. They have a lot of support for the cores. I am hoping that a lot more universities can do this. People wonder how to get the money that they need. Well, you can partner with industry, you can write instrumentation grants. How about hiring some folks to write those instrumentation grants so that you can be in the lab getting the data? Thinking outside the box, how can we do this? I need to skip some of this. Weaknesses of the core. This is, I'm preaching to the choir here. Cost of maintaining it, cost of buying it, cost of your staff to educate folks, cost of software, cost of the liquid nitrogen that you're feeding it, all of these things. Markus told us a lot about the prices and how he costed his out. The same thing is true for cryo-EM. Training is time consuming. I've heard some people say you need to get the graduate students and train the graduate students and they'll run your cores. I won't even let a graduate student touch these things, much less run them. So I think that the administration has to understand that some of these models are not a one-size-fits-all for all the cores. JBT had an article recently about shared research resources and what they recommend. You can take that to your administrators. ABRF has a resource page where you can find facilities that have self-identified. Even through some of the vendors I found, they have a page where they will tell you different facilities that have different equipment. At some point, your university has to decide whether they're going to buy their own or whether they're going to send you to a national cryo-EM center. 2016, 2017, NCI did a pilot study. They created a model national center. You send them a proposal, they agree or disagree, it's free, you get accepted, they take your sample, they run it. There's a video conference while they're running it so that you can see what they're doing and interact with them. That all worked out very well. There are now national cryo-EM centers. They have a particular mandate that they have to operate within, and so some of that is good for a lot of us because we can send our users there. Some of it doesn't work for some of the projects where they need iterative sample runs, they need more help with the experimental design, they need more help with image analysis. So that's where some of that comes back to the cores, and so there needs to be just a collaboration all the way around. Lastly, just some future thoughts to throw out there. Deep learning, how to apply different algorithms to those image processing, to the images to get your cryoparticles out of there without having to do tens of thousands of things, without having to do days and days of running on the microscope. I charge, in some cases, by the project, in other cases, by the beam time. Again, I try to put the staff time into that fee so that I'm not charging them a staff fee as well. And then I have no idea how much time went by, so I will leave it there. And cryo-EM, apparently I could have talked for the whole hour. My apologies.
So
we'll keep going.
I'm just going to switch with it.
Yep.
If you have any questions for Chris while I switch, please feel free.
Yes, sir.
I have a general question. So I come from a cryo-EM background, but I now run a protein core.
Okay.
It's broad in the sense of from isolation to characterization, my physical stuff. And I'm wondering how many people, as cryo-EM or any techniques, as your user base broadens, I know from my own experience, sample quality is everything. And so how do you interface with that challenge, especially if people are coming from non-biochemical labs? And maybe I'm just making a plug for like groups like mine that, we're upstream of that and as a model for making the most of the time on the instruments.
So I would totally agree. That would be certainly a plug for core facilities. Very rarely do you have a PI who knows everything from start to finish. So I think that the project meeting basically says, okay, if you haven't talked to the protein people, you should go talk to the protein people, because there's so many things that can go wrong that would waste your time and money. Most of the PIs go, oh, okay. So yeah, plug for the course totally. Take more notes.
Why don't we wait to the end just so that Soma has a chance? Thank you.
Can you want to reset the time?
Can you reset the?
It's doing
Dylan?
whatever it's doing.
Yeah, it's on its own mindset.
I totally got lost.
I use this to advance.
You should be able to.
Okay. Hello. Thank you for showing up for the structural biology talk. We are really pleased to see all of you here compared to yesterday we had a session. Before that, I really want to thank John for being the chair and organizing and getting us all in one place and reminding us when we have to stop talking, because I can't keep talking. My talk is going to be slightly different because Markus and Chris went through the details of the core facilities. You are not going to see instruments pictures or how to solve the structures. I guess LDA that you know it, or I'm not going to talk to you about it. Because it's an ABRF conference, mine is more like the challenges, how a core facility runs and how we. I thought I had the longest term in my facility. Markus beat me by 1 year. He joined in 1992. I joined in 1993. I've been running this for 31 years. Therefore, I'm going to share some of the high-level talk on the challenges. As I said, this is a kind of outline. I'm going to, already they've talked about the structural biology course, and I'm going to give a historical perspective of my core, X-ray crystallography core, and some status as challenges, and then how are we repositioning, or whether we are even not repositioning, like Markus asked, whether are we going to be there. Therefore, as I said, do you already have seen some of us talk? That's coming from the third person to talk. You are kind of has been covered everything. Therefore, you don't have to say some other things. As you know, the structural biology course are probably unique for an institution. You probably have only one, simply because it's very expensive, and also there are not that many users on this facility. They're very niche course. They are not the regular course you do in biology or chemistry or physics. We are niche course, and then usually there's one per institution, and then we come as staff people, we come with diverse backgrounds. I was trained as a chemist, and then I saw the light in biology. I became a biologist, right? That's how we come. And then some of us have physics background, engineering background. That's what we are, because that is what is needed to run a structural biology course, because we are truly interdisciplinary, right? And then our software are more open source, and then you should be comfortable running code, writing scripts and things like that. And then your hardware, there are very, very few vendors. And of course, everybody knows about service contract. I don't have to preach to a choir of ABRF people about service contracts. Therefore, we already talked, and Markus and Chris came to this topic. I run a home source. I'm not going to show the pictures of my X-ray instruments because it's no longer going to exist, because it was bought in 1993. Now we are in 2024, that instruments are no longer relevant, right? And then national facilities and regional centers are actually taking over, making my either life easy or miserable, depending upon how I look at it, right? Or they are going to replace me, I don't know. As I said, there are X-ray crystallography. I live in Tallahassee, Florida. We have one of the unique things which Markus would love to have it in Vanderbilt. We have the National High Field Magnet Lab, right? We are the biggest magnets in the world, right? We have a national facility. Therefore, we are going from a small regional core facilities to either regional facility or national facility. That is the future of some of the cores, right? And I'll just touch upon that. And then, as I said, we are an interdisciplinary person. This is both goes for the people who use it and the people who are the people who manage it, right? You need to have the molecular biology skills to clone, express the protein. I'm talking mostly about proteins, but it doesn't mean it's only proteins. It could be any biomolecule, right? Lipids and then combination of nucleic acids and proteins. You need the skills, right? Therefore, we attract students who come with a strong molecular biology skills, but they also need to have some biochemistry skills to purify the protein, make sure the gels are the samples are pure enough to crystallize, and then comes to the physical process. After all, you are dealing with the electromagnetic radiation interacting with the biomolecule, right? People have to have an idea how those interactions work, how that is going to impact them. You know, when you go to a dentist, you're going to put a lead apron, and they're going to kill you, right? I mean, without that, right? We are killing the samples with the X-rays. Therefore, the sample you start with may not be the sample you see after 2 hours, because we are bombarding with a very high energy X-rays. And that's why about 1995, by serendipity, somebody froze the sample in a loop, and then our trajectory changed. We collect everything at 100 Kelvin now. That became the lifesaver for most of the macromolecular X-ray crystallography people. And then obviously, you need to have software skills. Most of our things runs on Linux operating system. People need to know how to write scripts, how to fix things. Some of them come with that background, but they don't have any idea about molecular biology. Therefore, as core facility people, we need to sit and talk with them, accentuating their skills and help them learn the things which they may not have, but eventually they had to have a PhD, therefore he helped them. And sometimes we actually behave like a psychologist, listening to the student come in crying, You see, my PA may not know that I don't know these skills, but we thought, okay, don't worry about it. We'll take care of it. We'll train you. That's the training component these two people touched on. That's very important. Right? That may be my life-saving, job-saving skill I have to have. And as I said, historically, when I started this in late 1990s, a student's PhD thesis probably will have one structure, right? This was done 3 years that person worked. They collected some few dozen datas. The datas were kind of 2.5 gigabyte of data. Oh, that's a lot of data, right? We used to back up in tapes, 8mm tapes. I don't know whether you know what a tape is. That's what we did. Now, a student's thesis needs multiple samples. They are not satisfied one, and that means dozens of data have collected, and data collection takes 3 hours instead of a week, and data collection now happens in National Lab. We send samples, we call it FedEx data collect. We freeze a sample, put it on a puck, send it to either Brookhaven National Lab or Oregon National Lab, and then they open it, put it in a bucket, the robot picks up the sample, collects the data for you. And then they send, not only collect the data, they process the data, ship the data through internet, right? Because everything is magic. And then the problem is we have a lot of data. I'm not wanting to say anything about the data size after Chris spoke about, because they have collect 50 gigabytes of data a day, and we are not that level yet, but we have a lot of data. How do you keep it? You may have heard somebody talk about fair and open. We are there, right? We have a lot of data. How do you do? I think Chris Lab probably pops in a 2 gigabyte or 50 gigabyte disk, keeps it on the desk. I can see my cryo-electron microscope people building stack up a hard disk, which has a whatever amount of data. How do you keep it fair and open? We don't know. That is a problem we have to deal with, right? See, this is 3-hour data we recently collected at our Brookhaven, right? You can't even read the names of the samples here. That much of data we are collecting. Imagine the amount of metadata a student needs to put in to keep it track of. They don't even know what data they're collecting. That level of data we are collecting, that is another problem we are facing. And this is the protein data bank, the people who are familiar with. This is where after 2 or 3 years of working on a particular protein, you solve the 3-dimensional structure and deposit the coordinates, the atomic coordinates to the protein data bank. And the EM people have the EMDB, we have the PDB. You can see that increasing an amount of data going in. And the other interesting thing is, I don't know whether people are familiar with this, the Google's DeepMind AlphaFold. Everybody is trying to make our job completely irrelevant, right? This is basically, you remember the biggest paradigm in molecular biology is, if you knew the sequence, you should be able to predict the structure. We existed this long because they couldn't be done, right? They came to Markus and Chris and me, try to solve this. Now Google comes along, they spend billions and billions of dollars and get up with this DeepMind. They can just solve everything. You don't even need to go to the lab. Everything is done, right? But that is the challenge we are facing. It's a slightly different challenge. That's the challenge we are facing. You can see all the human beings done some amount of structures, and the 1 million structures, all the proteins existing, they solved it. We don't know whether they are right or wrong. That is why we still exist. Will the NMR be there? Will the X-ray be there? Will the cryo-EM will be there? Probably will, because there are some of them we need our help. Okay, we talked about some of the advantages. I'm going to say some of the challenges we face. I said we need to upgrade equipment. I'm not going to talk about how we fund it, how we cost recover it, because these two people talked about some of these things. That is a challenge. And then we can supplement our skills. Some of the things I'll talk are with collaborators' course, or just simply merge your core, or just disappear, go to sunset, go home and have a retirement, happy retirement. This is one of the challenges. This is my beam, right? This is a synchrotron beam. This is 10 folds, right? They are about 10 million times more powerful than I am. They have more flux. How do I compete? There's no way I can compete with them, right? I don't want to show all the problems, but that's one of the problems. And the other one is, this is the smallest beam I can provide, 70 micrometers, right? The synchrotron gives you 5 micrometer beam line. Therefore, samples are becoming smaller. You really do not have to wait for the crystals to show up. It's almost like doing micro electron diffraction, right? And there's also serial crystallography where you can actually have a stream of microcrystals go through and the X-ray shoots it and collects all the data, right? We can't compete. Home source, home core cannot survive in this kind of a challenge, right? The other thing is auto-centering. Everything is automated. FedEx data, like we send the samples, we can send up to 17 into a puck, and they can send 6 puck. We are talking about 100 samples. They open the hutch door where the X-rays are coming due to the safety reason. They close the door and put it, and the robot takes. I want puck number 1, sample number 17. It goes there, it puts on the beam, you collect all of them. Everything is automated. And we sit at Tallahassee and control the beam line in Chicago, right? During our time. Everything is automated. I can't compete with that, right? That's difficult. The only component we survive is training. And then this is even more cool. You put your sample, because it's a frozen sample, like Chris gets into this, you really do not know where your sample is sometimes, right? What you see here, this is basically a loop, maybe about 1 millimeter move, and you see the crystal sitting right there, right? Here, for whatever reason is freezing is not good, you don't know where your sample is. They have come up with ways to put the loop. It does up and down fast, taking microseconds of a beam going through your sample, and then you can probe where your sample is going to diffract the best. It's not necessarily the center of your sample, it's somewhere else. It reports to you. Therefore, we used to spend hours looking for this. It just does it in 1 second. And then we collect data. How do I compete with them? You can't. It's very hard. Basically, I'm saying I'm sunsetting my core. That's the final thing. Okay, there are also some opportunities. I don't want to be too depressing on a Tuesday afternoon or morning, whatever. Because of the structural human structural genomics, we have a lot of automation, lot of instrument developments, all kinds of miniaturization, automation, everything came because of that, right? That is a positive. Therefore, we still are needed because we need to do the data processing. We need to do the data in interpretation, right? You know whether it's right fold, right structure. And then finally, project involvement. Because things are going fast, they need some kind of hand-holding for you. The students find it bewildered by this level of speed in which they deal with it. Therefore, they come to you. That's where you are. Somebody asked whether we charge for the training. Obviously, I work in a university. Education is an important component of what I do. That's what I love doing it, and therefore, that's important. We don't charge for it, but we do that. And then sometimes we ask help experiment design, sample preparation, sample storage. These are the things we try to do. That is still some opportunity we have. And then I try to expand the services I give. Because for me, if they don't crystallize, they won't come to me. The protein doesn't crystallize, they won't come to me. Because my home source is shutting down, how do I expand my service? I provide crystallization robots. I help them, they just bring the protein. We test out multiple screens like Markus was talking about. There are commercial screens. That means screens means different kinds of buffers which a priori they do not know the crystal will and the protein will crystallize. We try thousands of different commercial different buffer systems to see whether it will work for them. We provide that service, and then we can make custom buffer, and it can go from a particular concentration, 10% up or down, different pHs, different concentration, multiple components. We can make custom buffers so that what used to be a microcrystal or a precipitate can become a single crystal so that they can collect data. Of course, we provide services like Chris talked about, storage and shipping, data storage, and backing up, all those things we help them. Other possibilities are, I have to bite the bullet and merge with another core, right? there's one final possibility if I don't. How can I do that? Because some of the skills I've learned over the 30 years probably still relevant in small molecular crystallography, right? Some of them may be useful in powder X-ray diffraction, which is in material science. People still do that. Maybe I can leverage my experience and my knowledge in going there. It's not necessarily always possibility for me, but at least there. Or, as I said, material, otherwise, electron diffraction. Some of the techniques are different, but at least you have the background, physical interaction of matter with biological molecules, and some of the mathematics going behind that. You have some idea, therefore you could maybe find a job there or something. Everything else, sense at the score. What else you do? I mean, it is a depressing. I don't have a depressing end, but that's one of the possibilities we have. That's why the sun is here. Think of it not a sun-setting score. It's actually sun is rising there, right? You can interpret it whatever way you want. And, as I said, to summarize, some of the topics I talked about, the challenges are rapid changes in automation, technology, and efficiency. We can't really do. We are adapting. We have to, as humans are being pushed to the wall, you figure out to do something. You try to claw up on the wall or something like that. Then some of the things that we talked about, we can expand the services. I can update my skills. I didn't talk about here about the training. At Florida State, what we have done is we have what is called a boot camp, where when the students come, we teach them in 3 weeks at various core facility how to do what we do. Therefore, even before they begin their PhD, they have an idea. We do it for free. We select simple projects which cut across all the cores. I mean, my colleague here, Peter is here. He runs another core. They will be in the Monday morning with me. Monday afternoon, then he goes to Peter and he teaches what are the instruments and core skills he has. Next day, they go to my other friend, Guvi Moon, who does the protein purification expression. And they come back to Wednesday when the crystals are ready, they go through. The training is becoming, rather than the charge part, it is an important component of why we are still relevant. Otherwise, I said we can merge the score or census score. I hope I did not run out of my time, but I think I've taken 18 minutes. I'll be willing to take questions or take questions from everybody. Thank you very much. Appreciate it. Thank you, guys. Thank you. Yes, Josh.
I have a question for the panel.
Okay, go ahead.
Maybe this is better inside conversation, but I thought I'd
You should go ahead. I think we still have time. Yeah.
What are your thoughts on national centers and how does your core utilize national centers as a resource? In each technique a little bit different. The national center models may be a little bit different, but I think there's a lot of common threads between each of the different technologies or the cores.
Let me address from the X-ray part. As Chris pointed out, we have 5 centers, kind of 5 or 6 centers, one in Brookhaven, one in APS, one in SSRL, and one in ALS in Stanford. That is the model invariably coming because of some of the challenges and competitions we have. The way in which how the NSLS2, National Synchronous Light Source 2, does is, they are writing a grant submit and then they have to be justified. Right? They push most of the scheduling, everything to me, right? Therefore, I am their staff, even though I don't work for them. We are given what is called block allocation group unit. They will tell me on Tuesday on March, whatever date, you get 8 hours of beam time. You figure out what you will do with it. That becomes my responsibility, get all the PIs in my campus to find their time, coordinate with the students. 1 week earlier, 10 days earlier, we do the safety forms and we do the sample preparation, sample shipping, all this management I do 1 week ahead of time. And then on the day of the sample data collection, we sit and become 2 huge monitors, log in through no machine connection, and then run. While running, most of the students are still, especially if you do not see what you are doing, it's actually more complicated, right? You say, add the samples in the goniometer. What is a goniometer? They don't even know what it is. They've never seen one, right? This is what's happening to the new generation of students. It looks like a magic box to them, but that's inevitable that is coming, right? And then it's free. How do you beat for that? There's no service contract, there's no maintenance, there's no data back, no, you don't have to worry about any of that. That's coming whether we like it or not. At least in my case, that is the future. We use that because of the efficiency and the faster this happens. It's inevitable, at least for the X-ray case. Yeah. Go ahead.
So my experience is in the 90s, we were actually going up to Madison using that high field facility up there. And we were just basically running samples day and night. The one challenge with NMR is that often our samples are not long lasting. And that's why it's a little bit tricky to ship them around, to travel around with them. And that makes it kind of hard. And what I see with some of these national centers is that whoever has it has to benefit. And then all the others in the consortium kind of struggle to actually use it because of sample prep and sample stability, viability. So it is a challenge, and I'm not sure if we have to move to that because of the instrument cost, how it's going to work out.
I would just add that if you're just a regular EM lab, or you're just getting started and you have somebody come to you with a project, your best mode of operation is to tell them your limitations and to tell them about national centers, and they decide, and then they have that experience. And in some cases, it's very good for them, right? It's free. I've heard that the queues are very long, so getting your time on the scope. The National Centers for Cryo-EM have a very narrow mandate, and so the things that they can do and offer you are limited. So core still has a role in that, sample preparation, data interpretation. There are certain schools and universities that are putting forward the money to make cryo facilities, and they need to understand their users. There is a tipping point where it makes sense for you to create such a thing. One of my colleagues is heavily invested in that at their university. It is subsidized in a lot of cases by external users, pharma, different places who are coming to use their stuff. So that allows them to pay their bills and offer it to the PIs on campus as a more affordable service. So I think it has a role, definitely. It's going there. There's no stopping it. Money is driving this.
Yes.
How do you manage the kind of cost associated with instrument downtime, especially for the particularly expensive instruments that you don't have duplication?
I'm really fortunate. It may come as a surprise. In the 30 years, I never had a service contract. Right? I don't know whether it is because the instrument manufacturer had a good run, or because I come from a state university, we have machine shops, glass blowers. I am an employee of the state university. You don't have to pay me to do anything, right? Regarding going back to the teaching, right? You don't have to pay Peter to be there, right? We have glass blowing facilities, machine shop, electronic shop in my unit. It's going away because now that we are using and throwing kind of culture we have come. Sorry about I'm an old person. But my PhD involved putting an instrument together. That's what I built my instrument because the commercial instrument was not available. Markus would tell the exact same thing. We were tinkerers and fixers in our earlier incarnation. Therefore, we are fortunate enough. What does the service tech do anyway? He finds out which PC board is not working, throws it out, puts a new one, right? We are all afraid. Of course, I'm not talking about Chris because there's a $5 million equipment. If you touch it, the service person say, no longer warranty is valid. We are afraid of that. At least we took some risks, right? Okay, so what? We will try to fix it, and then that's what we did. And go ahead, Markus.
I think it's a very valid question, and we struggle sometimes with it. Different than Soma, we actually have our salaries are inside the fees, so we have to recover our fees since we are in a private institution. But the key in my mind is to minimize downtimes. And minimizing downtimes, in my experience, can happen by training and some supervision. So I tell my users the expense of the instrumentation, the impact it has if a cryoprobe goes down, that it may be gone for everybody for 3 to 4 months. We've had one out for 2 years. And so it impacts everybody. And that's kind of when I not scare them, but I tell them the impact and that I'm here to help them. So come to me if you have a question, if you're uncertain. I'd rather be bothered and help you than have to deal with the aftermath.
I would agree. As a core director, the amount of time I spend sitting in my chair is minimal because I pop up every time somebody walks in, has a question, has a problem. I am better served to address it before it becomes a problem.
Right.
When a machine goes down, and they do, service contractor, no. Human error or no. I have a working relationship with the users such that they know I'm going to tell them straight up that it's down. They know I'm going to get it fixed as fast as I can. And they know I'm going to tell them what the problems are. And as long as they're kept in the loop, they've been pretty understanding because it's out of my hands. So you manage it because it comes along, but I would agree with Markus that a lot of it comes down to the training and the supervision. You can prevent a lot of things.
Yeah, actually, slightly a different take on that is because, I mean, we are a course facility manager. When the new students comes, I'll tell them, okay, I'm going to train you, and I want you to call me before you use it. I put it this way. If something happens, it's on my facility's dime. If you do it without knowing, properly trained, you try to do it, it's your PI's dime, right? You should, I mean, I'm not actually answering your question directly. And then because we are core facility manager, like Chris correctly pointed out, we are there, and because we see this in PI managed equipment, there's nobody. PI doesn't do it, students don't do it, postdoc don't do it. They come do it, their experiments go away. They don't even clean after themselves, right? Whereas because we are always kind of legal, we are looking at it, even becomes a big problem. Eventually it will become, I'm not denying that you have to find, there's downtime. You do it, and like she said, I inform them even if it becomes a problem. All my users, look, there's a problem. We have to have some downtime. Please collaborate with. Communication is very important, like Chris pointed out. You tell them, they understand because they have stuff, breaks, their samples don't work. You communicate with them, tell them ahead of time, give them an estimated time for repair and reback. It's not necessarily the time you want, but at least they can plan, especially like Markus said, some of the samples may not be stay for a long time. Therefore, at least they can plan their preparations and then their life around it. Eventually, it'll break down, but at least minimize the problem. Yeah.
Okay, well, we're out of time, but let's thank the panel again, and I'm sure that they're willing to chat some more if anyone has any questions.